Hello, everyone. So my name is Dr. Vivek Tripathi, and I'm currently Associate Professor at Nooks and Nite University in Karnataka State. So in this brief presentation today, I'm going to speak about that how the breast cancer research and the CRISPR-Cas9 technology is actually transforming the innovations and cure to improve the lives of cancer-suffering patients in coastal region of Karnataka State. As we all understand that today the breast cancer is emerging as a serious public health concern across the globe in 21st century, including India. Moreover, when we look at the statistics of the WHO report for top 15 different types of human cancers which are found in Indian population, the breast cancer is on the top. And particularly when we look at our NCR report, it indicates that the breast cancer incidence and its related deaths are found all across the different states in India. However, its rate is steadily rising in urban as well as the rural areas, and that is actually a concerning matter, which is highlighting the need for development of much more sophisticated therapeutic strategies for the disease management. So the question is that why exactly this breast cancer is spreading so fast, particularly in young females? And the reason being, one of the reasons it could be the genetic factors. For example, the breast cancer causing genes, mutations in BRCA1 as well as the BRCA2 can actually have a lifetime risk of developing the breast cancer. So therefore, sometimes the genetic screening of those particular families can actually help in disease management. And there are other, the major factors are such as the no breastfeeding, and sometimes the alcohol and the cigarette consumption is also contributing, particularly in youth females, as well as unhealthy diets and use of the contraceptive pills. So these are the different reasons which are actually contributing in cancer development. Now, the second problem, which is actually the researchers are facing, that why are we not able to develop one unique therapeutic strategy for the treatment of breast cancer patients? And the reason being that this is such an heterogeneous and multifactorial disorder that actually can vary from one subtype to another. And this is the reason that according to the current breast cancer and research diagnostics, we need to identify novel therapeutic strategies and to develop precision therapy for every individual cancer patients. So in alignment of this vision, actually, we have proposed one research project under this PM ECRG from this Anusandhan Laser Research Foundation, and which has been approved and granted. So during the first year of this research project, actually our vision was to stratify all the regional breast cancer patients which are coming and admitted to our Justice K.S. Hegde Charitable Hospital of Nite University. And the first year of our statistical data indicates that approximately 45 to 50% of the breast cancer patients who were admitted to our hospital were carrying actually the luminal A type of breast tumor. And surprisingly, in our all the cellular studies, in most of the cases for the basic research, we use these luminal A like MCF7 and other cellular models as a control counterpart with respect to the other TNBC and other cell lines. So therefore, we are actually adopting a new methodology or a new way by which we are choosing much more physiologically relevant cellular models to compare and identify the novel diagnostic markers which can be used for the early detection of the breast cancer in our regional breast cancer patients. So therefore, we are actually collecting these human biopsies and trying to analyze the expression profile of certain SOMO enzymes as well as the FORC stabilizing factors, which can be exploited as a novel therapeutic targets. In parallel, actually adopting a genome-wide studies are being indispensable for the development of next generation therapeutic strategies for breast cancer management. So in that regard, we are actually performing a genomoid transcriptomic analysis to identify the novel factors for early detection as well as for the combination therapies. And in parallel, recently the fork stabilization through the fork reversal process has emerged as a novel cellular pathway which is contributing in the chemotherapeutic resistance and limiting the therapeutic efficacy. So therefore, we are, after the identification of the novel factors, we are trying to study that how these factors are contributing in the FOC stabilization and subsequently limiting the efficacy of these chemotherapeutic drugs which are being used for the treatment of cancer patients. And in parallel, the synthetic lethal interaction studies are also another arm where the researchers are focusing. So in that regard, we are using this CRISPR-Cas9 technology to generate basically the knock-ins and the knockouts, and then employing the latest DNA fiber technology to study that how the FDA-approved chemotherapeutic drugs, particularly the PARP inhibitors, are affecting the therapeutic responses in patient-derived cancer cells, which can actually be used for the clinical decisions and to design much more precise therapeutic strategies for the breast cancer patients in the coastal region of Karnataka state. So this is just a brief overview of the laboratory research work, which is actually contributing in the field of breast cancer research and diagnostics in India. Thank you very much, sir.